Hi everyone. In this presentation I will provide the background for the project I will be undertaking as part of my Masters by Research at UWE. I will be focusing on the biological role of the mitochondrial branched chain aminotransferase protein BCATm. I will be looking specifically at the potential role of BCATm in mitophagy and whether abnormal BCATm function could implicate the protein in Alzheimer's disease by inducing deficit in mitophagic flux. Okay, so to explain the relevance of this research into Alzheimer's disease, here is some information on dementia, which is an umbrella term for a key group of neurodegenerative conditions. Dementia is defined as a progressive clinical condition or syndrome that's characterized by decline in multiple aspects of cognition, enough to affect daily functioning. There are numerous disease pathologies that fall under this umbrella, as mentioned. It's a key group of neurodegenerative conditions. Global prevalence is estimated at around 50 million patients, and this is expected to triple within the next 30 years. As such, the disease presents a large socio-economic burden globally. Current therapies aim to slow progression of these conditions, but currently none of them reverse any of the pathologies involved. On the right here we see figure 1, which is the kind of main pathologies under this dementia umbrella. So, the most common cause of dementia is Alzheimer's disease, often shortened to AD. AD is characterized by short-term memory loss alongside personality changes. These behavioral changes are associated with pathological loss of hippocampal and neocortical neurons. The pathogenesis is complex and remains poorly understood. The primary pathologies are thought to be abnormalities in neuronal amyloid beta and tau function. However, the condition also presents with glutamate toxicity, alongside dysregulation in other cell functions such as metabolism, autophagy, and protein folding. Research into potential links between these pathologies is important in elucidating the full pathogenesis of the disease. On the right here we see in figure 2 an MRI of a healthy brain versus an Alzheimer's disease brain with clear macroscopic changes in brain structure associated with loss in these neuronal types. Perhaps the most well-known focus in AD research is the amyloid beta tau hypothesis. Amyloid beta and tau are proteins with important roles in neuronal function, and a hallmark pathology noted in AD brains is the appearance of abnormal neurotoxic amyloid beta senile plaques and tau neurofibrillary tangles, SPs and NFTs. Initially, it was suggested that aggregation of abnormal forms of amyloid beta initiated NFT formation. However, later research showed that SP and NFT formation are separate events. Furthermore, anti-SP and NFT drugs did not reverse AD in mouse models, suggesting a more complex pathogenesis is at play. And figure 3 shows the suggested hypothesis between amyloid beta aggregation and tau NFT formation. So, looking more closely at one of the cellular processes thought to be dysregulated in a range of human conditions, including AD, we see that autophagy under normal physiological conditions functions to recycle damaged or dysfunctional cellular components, maintaining normal cellular function. There are a range of triggers and a range of inhibitors. We see that it's clearly quite a complex process. So a trigger such as stress can initiate the autophagic cascade, which results in recycling of cellular components, preventing the cell from incurring too much damage. Next, we look specifically at the role of autophagy in the brain and linkage with neurodegenerative conditions such as AD. The brain is a highly metabolically active organ and remodels via synaptic plasticity. Autophagy is vital in maintaining this ability to remodel. Deficit in autophagy is associated with neurodegenerative diseases, and impairment in synaptic plasticity and reduced autophagic flux are observed in AD models. Autophagy is also implicated in modulation of amyloid beta function, so presents an interesting focus in AD research. Mouse models demonstrating impaired neuronal autophagy exhibit a higher intracellular amyloid beta load. Figure 6 shows an overview of the proposed signaling pathways involved in autophagic control of A beta, showing that abnormal function could lead to amyloid beta toxicity and buildup of extracellular amyloid beta. Even more specifically, we look at the potential role of the protein BCAT, Branched Chain Aminotransferase, in autophagy and thus AD. Ordinarily, BCAT exhibits an important metabolic role, seen in Figure 7, catalyzing the transamination of amino acids. It is also neuroprotective, initiating autophagic A beta clearance. The protein exists as two localized isoforms, mitochondrial and cytosolic. Under pathological conditions, the cytosolic isoform, BCAT-C, shifts to a neurotoxic role. Intracellular changes increase BCAT-C translocation to the autophagosome membrane. This increases autophagic flux, causing intracellular accumulation of amyloid beta containing autophagosomes. This prevents lysosomal degradation of amyloid beta. When we compare both isoforms of BCAT under pathological conditions, we see that both exhibit regional increases in AD brains, implicating both in glutamate excitotoxicity. In other dementias, BCATm is increased at physiological BCAT-C expression, and disease-specific BCATm mutants have been observed. This suggests that the two isoforms play independent roles in pathogenesis. Whilst the basic role of BCAT-C in autophagy has been characterized, the role of BCATm has not. Looking at mitochondria specifically, we see that mitophagy is the selective autophagic removal of damaged mitochondria. Mitochondria are intrinsic to vital cellular functions such as bioenergetics. Accumulation of damaged mitochondria as seen in AD, implicating mitophagic deficit in the pathology of the disease. The mechanism of mitophagy is complex and not fully characterized, though defects in mediators have been seen in AD models. This leads us to our primary question. If BCAT-C regulates autophagy in the cytosol, does BCATm regulate mitophagy? If so, could dysfunctional BCATm signaling explain AD mitophagic deficit? Okay, so on to the aims and objectives in this project. The primary aim is to investigate whether BCATm plays a role in cellular mitophagy, and whether impairment of BCATm function could be mechanistically linked with mitophagic deficit observed in AD. The primary objective is to induce knockdown and overexpression of BCATm in a cell model using short interfering RNA, siRNA, and DNA transfection respectively. The HeLa cell line exclusively expresses BCATm isoform, so is an appropriate model for this purpose. The secondary objective is to measure mitophagy markers such as DRP, OPA1, and mitofusin. Comparing differential levels of these will allow us to establish whether knockdown and overexpression of BCATm has an effect on mitophagic flux in these cells. In Figure 9, you see a general representation of mitophagy and how the markers DRP1, OPA1, and mitofusin can be used to figure out the levels of mitophagic flux happening. Thank you for listening to my presentation. I hope everything was clear. If there are any questions, feel free to leave them in the comment section below. Here are my references as standard. Once again, thank you for listening.